Hi guys, just a short video today to talk to you about inheritance to help you answer your questions this week. So the first thing that we'll go over is just easy stuff. So the difference between meiosis and mitosis. So there are a couple main differences, right? So remember our PMAT? Right, so our prophase. And then we could have cyto at the bottom. Right, so we could have cytokinesis, but our PMAT, right? So we have prophase, metaphase, anaphase, telophase, right? So this is mitosis over here. So you notice with mitosis, from where we start is the same as when we end. So mitosis produces clones. So if you look at meiosis, right? So we have prophase 1, right? We have metaphase 1, we have anaphase 1, we have telophase 1, which they all show in this picture, right? So they kind of group these together, telophase 1, anaphase 1, and telophase 1, right? But then at some point, we'll have prophase 2, metaphase 2, right? And then we have cyto, right? Cytokinesis. So you'll notice that now I have 4 daughter cells, and each of those are different from where we started, right? So each of those are different from where we started. So let's go over some things that are important. So let me erase this. So I forgot what I was going to say. So mitosis creates clones, right? Meiosis creates genetic variation. So that's the two big differences between them, right? So this creates clones, and this creates genetic variation. Okay, so important things to remember on how it happens, right? In prophase 1, I get crossing over, right? So at prophase 1, I get crossing over. So kind of important things. Remember some important, so if I have my P1 here, right? Here, I get a tetrad and crossing over. And that exchanges pieces of DNA. So at the bottom here, right, I get sisters or daughter cells, and they have different combinations of chromosomes. So the maternal and paternal chromosomes get mixed up, right? And that's what it talks about here, right? So that happens in prophase 1, right? Another thing that happens is independent assortment, right? So I've got my independent assortment here. Right, of chromosomes. So independent assortment happens at metaphase plate. So this right? So what that says is that all the maternals and paternals don't line up, right? It's mixed, so I can have maternal and paternal, and the next one I can have paternal and maternal. So you'll see here that I have different combinations of what can happen. So that happens at metaphase. It happens twice, right? So we get independent assortment twice. So, but for most part, metaphase 1 is the important part. So we'll put that here. Metaphase 1. And then genes can be linked or unlinked. So genes on the same chromosome are linked. Genes on different chromosomes are unlinked. So that's a brief overview of that. That's probably all you need for your questions. So inheritance patterns. Right, so if we say so inheritance patterns, we've gone over these a lot. So dominant inheritance patterns, autosomal dominant. So autosomal, remember this. I'm stuck. Okay, hopefully that's better. So remember that autosomal means non-sex cell, right? So autosomal dominant says if mother has a dominant allele, remember we have 2 alleles for each. Two alleles for each trait. So if mom has a dominant trait on one of her alleles, she'll equally pass that to both males and females. If dad has a dominant allele, he'll do the same thing. So my chance, if I have a dominant, is 50%. If I have a recessive trait, so remember autosomal recessive, right here. So autosomal dominant, I only need 1 copy. So autosomal recessive, I need 2 copies. So if mom and dad are both carriers, right, 25% of my offspring, if you remember your Punnett squares and stuff, 25% of my offspring will have the disease. 50%, right, will be carriers. I guess they had it over here like this. So this is my affected child over on this side. Right? And then 25% will be unaffected. When it starts to get different, we get to sex-linked. So sex-linked, right? Really, we're talking about the X chromosome. Not a whole lot on the Y, so the X chromosome. So if I have, in this first situation, if I have a male who has a disease like hemophilia, which is sex-linked, the chance of him passing that disease to his offspring is around 0. I'll say around because there are unfortunate X inactivations we've talked about, but near 0. So he'll pass the X. He can't pass the X to his sons, but he can pass to his daughters. So if the male has it, then all daughters. I can do daughters easier. So all daughters are carriers. So if I have a female who's a carrier, that's all sons. All sons have the disease. And then 50% of daughters will then be carriers. Because 50% will get a disease, and 50% won't. But all the sons will get the X. Right? So just remember that. And the other inheritance that is not talked about as much is mitochondria. Important things to remember about mitochondria. So these are inherited. So they're inherited in egg cells. Not in sperm cells, but in egg cells. So they're all inherited in egg cells. So what that means is if mother has a mitochondrial disease, then all of her children will, because all of her children got her eggs. If dad has a mitochondrial disease, none of the children will, right? Because the sperm carries no mitochondria. It has some in the tail, but the tail gets chopped off, and it doesn't become part of the new genome. So if we think about mitochondria, by the way, if we think about mitochondria, so if I have a mitochondrion, right, in the mitochondria, remember, these are our endosymbionts. It has one chromosome. So it is 1N. So there's no recombination, right? And these reproduce by binary fission or budding, and it produces more mitochondria, which are clones. Right? So all the mitochondria will be the same. It's unlike up here, where all of these situations up top, with people, all the people, are 2N. I lost my... all the people up top, 2N. All right, but the mitochondria is 1N. All right, last thing I need to tell you about really quickly. You'll be asked to draw a graph with error bars. So there's a type of bar graph called a bar whisker. And these whiskers, or what they call standard errors, right, or what we call standard deviations. So each of these little things at the top is a standard error, right? So you'll be asked to draw a bar graph this week, and it'll say include your standard errors, and that's what they're talking about. So the grader wants to see that you have both bars, and you have these little error bars inside. So if you're got my y and my x. So 1, 2, 3, 4, 5. If my standard error is equal to 0.5, then I simply include a 0.5 error there. So if that's what's asking you to do, I just want you to be prepared for it. All right, well, good luck this week, and I will talk to you soon. Bye.